All right, so today we are going to talk a little bit about microbial control, which is, of course, a pretty important part of microbiology because a key thing is that many microbes cause us trouble, and we want to find ways to control their growth or not let them in certain situations. So, when thinking about microbial control in general, there's really sort of two categories of control methods that I'm going to talk about today. And then the second one breaks down further into areas that we're going to get into that I'll get into in another video in the future. So the major types of control methods are physical controls and chemical controls. So physical controls typically involve some kind of environmental feature. So for example, fire, heat, temperature is a physical control. Other ones include pressure, they include radiation, they include things like that. Chemical controls are, of course, chemicals that in some way either slow or stop the growth of microorganisms. And those chemicals come in a variety of forms. Disinfectants are chemicals we often use on surfaces. Antiseptics are chemicals we often use on surfaces that are living, like the human body. And then there are actual drugs that control microbes, and those I will get into in a separate video. This is going to be more about physical and chemical controls for various kinds of surfaces, not internal to living organisms. All right, so when thinking about microbial control, what do we want to think about? Well, we want to think about, if we're starting with the control method, what type of control we have, how it affects the microbial physiology. So are we talking about a certain aspect of microbes? Does it kill the microbe or only stop it from growing? Those are two different things, and it depends on what you're attempting to accomplish. Does it hit microbes in an overall way, or does it nail certain bits of their physiology so that some might survive and others might not? Which types of microbes are you attempting to control? Are you trying to kill bacteria, viruses, fungi? Those are all very different. And even within those groups, there are some that are killed more readily by some things than others. And then, of course, with control, there's almost always sort of a payoff or a downside. So I mentioned that fire or heat is a great physical control, but you can't just light everything on fire. You would destroy it. So you need to think about whether or not you're damaging the item you're working with, whether or not it's going to work well enough, how much of something do you need to destroy the bacteria versus how much is going to hurt the item. If we're talking about dealing with living surfaces, we want to know if these items are dangerous to us as living beings. So these are all things to think about. There are so many things we could go over in terms of control. I'm just going to give you two examples of things you might think about. So my first one is going to be hyperbaric oxygen therapy. This is a physical control method. What it does is a person goes inside a hyperbaric oxygen type of tank. It increases the oxygen pressure around them, which also increases the oxygen pressure and concentration in body tissue. So pressure overall doesn't usually do much to microorganisms unless you get to the point where the pressure is so high it would kill any living thing. But in this instance, the pressure is being used to increase the oxygen, and so it's a little different. Pressure is also often combined with heat as a method of microbial control. So in this instance, what the attempt to do here is to slow or stop the growth of bacteria that don't like oxygen. So aerotolerant organisms would survive, but probably wouldn't grow well. And we would call that bacteriostatic, as in it stops their growth and leaves them at a static level. But if there are obligate anaerobes, it could actually kill them. Oxygen is toxic for some bacteria, and they do not have the enzymes necessary to fix the oxygen radical problem. And then it might be bactericidal, as in it kills them. Specifically, hyperbaric oxygen therapy is often used for Clostridium. So Clostridium stridellii causes necrotizing fasciitis, which eats away at various tissues. And Clostridium perfringens causes gas gangrene, which does the same thing in a slightly different way. And both of these can be helped using hyperbaric oxygen therapy. It increases the oxygen concentrations. The Clostridium are not happy campers, and they do not survive. Even their endospores may not survive in those cases. So the downside, of course, is that we are not built for excessive amounts of oxygen or oxygen pressure. And although we are very, not just aerotolerant, but obligate aerobes, it does not mean that increasing the oxygen pressure is always good for all of us, and it can damage some human tissues that are very delicate. So you want to be careful in using these methods. All right, so now that was a physical method. A chemical method might include something like triclosan. Triclosan is a chemical that has been added to a number of substances nowadays. It is a phenolic. It has phenol rings in the structure. Here you can see the 2 phenol rings in the structure. It is considered to be an antiseptic, as in it can be used with human surfaces, but also inhibits bacteria. Now, I've got some question as to that antiseptic thing. It may or may not cause harm to humans, but that's actually not as important as some other aspects of it. But first of all, how does it work? Well, it blocks fatty acid synthesis specifically in bacteria. It blocks a pathway that bacteria have that humans do not. So it does not block our fatty acid synthesis, but it stops theirs. And bacteria need fatty acids for their lipid membranes. It stops lipid membrane growth, so it is generally bacteriostatic. It doesn't directly kill them, but if you can't make new membranes, you can't grow, you can't divide. So you'll eventually have problems. It's specifically used for things like Staph aureus and Staph epidermidis, both of which are typically on or related to skin, but cause various kinds of infections. And so in healthcare settings especially, triclosan was used quite a bit as a surface cleaner and things like that. You also find it in toothpastes, which is one of the Staph epidermidis things. It tends to go after teeth. You can also find it in deodorants and a number of other things. Hand soaps are still the most common. However, downsides, it seems that this particular chemical, while it may or may not cause actual damage to humans, definitely seems to increase antimicrobial resistance from the microbes around it that survive. So it seemed like it might kill all the bacteria. Turns out that wasn't true. I actually have a personal relationship to triclosan because a lot of the research was coming out about it when I was in college. And in my environmental microbiology class, we did an experiment. We actually took 2 dorms. So we had 2 girls' floors and 2 boys' floors in a dorm. We went to their bathrooms. We closed off their soap containers and we put in our soaps. So they either had a triclosan containing soap or a non-triclosan containing soap. And in the girls' bathroom specifically, we very much noticed that in the bathroom containing triclosan, there was many more bacteria we collected on plates that were then more resistant to antibiotics than in the bathroom that did not contain the triclosan. So we did see some of the growth of the antibiotic resistant organisms. And you might ask what happened on the boys' floor, and we didn't see nearly as much of a difference, but also we didn't have to replace the soap nearly as often. And so most of what we learned from this experiment was that college boys did not use soap very much. But either way, this has been shown through some actual research as well outside of my college experimentation. I did also get to run an N=1 study at one point with triclosan, as in it was really only about me, because my summer camp switched to all triclosan soaps the summer after I did that experiment at college, and I utterly refused to use those soaps. I went on a tirade. I brought my own soap, and I said I was not touching any of what they put in the bathrooms and that we should change all of it. Nobody listened to me. And that summer, we had a huge outbreak of impetigo. And impetigo is a staph-caused infection that causes lesions in the skin. And it's very contagious and it passes from person to person, but also it can be problematic in terms of antimicrobial resistance. And clearly this one was surviving our triclosan soaps. However, I never touched anybody or anything related to the triclosan soaps, and I never got impetigo. So I am saying that that worked for me. All right, but those are, again, methods of microbial control and how they work. What I want to do now is think through some mini case studies in microbial control. So these are all real situations that people are in regularly or that we have been in some large capacity where you might have to think about different ways to control microbial growth. This happens all the time. All right, what are the things we want to think about as we get through these scenarios? Well, we want to think about what are we trying to kill or inhibit? Are we going after bacteria, fungi, viruses, specific bacteria? What are we going for? Then what do we need to avoid damaging in the process, and how clean do things need to be and for how long? So both of my pictures here are ways we clean water in some fashion. Our swimming pools contain chlorine. We need them to constantly be free of bacteria, and we're trying to do so in a way that doesn't damage us. However, if you're trying to clean water for drinking purposes, perhaps you will just boil it. But as long as you don't drink the boiling water immediately, then it only needs to be cleaned for long enough that you're going to use it. and you're okay. So lots of different options, in this case, 2 just for cleaning water. So here's a situation that you probably encounter more often than you would actually think. We infuse plastics with antimicrobials. Not all plastics, obviously, but you can see here in this image of this child's changing table, this little blue thing here that says Microban means that these plastics have been infused with a somewhat proprietary blend of varying chemicals that are supposed to be antimicrobial. In the case of Microban, there's a very good chance that Triclosan is one of them, so I'm not a fan, but it doesn't really tell you exactly what it is. And then there are also some food products like cutting boards that have been infused in the same way. So you can buy antimicrobial cutting boards. All right, so let's pros and cons this here. First of all, you need to use the right kinds of chemicals if you're going to do this. These are things that are associated with children. Some toys have been made with Microban and other stuff, or food. We have to make sure that the chemical you're using is quite safe for human consumption. Again, I'm iffy on Triclosan. And also quite stable if it is going to remain in a plastic for an extended period of time. So you'd want to use something probably in the antiseptic range, but not something that's very volatile. So alcohols, for example, would not diffuse very well. They would be out into the air. On the other hand, I probably wouldn't want to put heavy metals in here unless I really know that they're not going to get out and into the people who are involved. So this is a pretty tough call. You want to have something that maybe stays clean without having to constantly wipe it down with some kind of disinfectant. But also, do you want to constantly kill everything that it comes in contact with? And there's definitely some generalized question about that and how that might work. And so thinking about both how the chemical affects the person and how it affects the area and stuff around them is important. So speaking of that kind of thing, let's talk about some of the different things we use to clean ourselves, like soaps versus antiseptics. So now soap is not an antiseptic. It is what is called a surfactant. The surfactant changes the way water and surfaces interact in such a manner typically to not allow things to stick to them. So if you think about soap, the idea is that it unsticks things from places, and in this instance, unsticks different kinds of microbes and bacteria that may have found their way to a surface. Soap is not meant to kill those microbes. It is meant to remove them from the surface, and then you can wash them off. So a big part of dealing with soap is not the soap itself. The soap should get on the surface, and then you actually need to physically move and scrub it to remove it. On the other hand, in some instances, you actually want something that will kill bacteria. Like this surgeon is definitely going to need to kill as many bacteria as he can if he's going to get inside of a person, wants to make sure that he's not dropping off any accidental microbes, including ones that would live normally on his surface, but not normally inside of something. So healthcare might get more into antiseptics. You want to kill more bacteria than you just wash away. However, don't forget your body is also covered in microbes, and many of them are an important part of our lives. Our microbial flora help keep us healthy, help keep us alive. So when you're thinking about washing your hands, which you probably do a number of times a day, you get your hands in something, you want to wash them, you go to the bathroom, you want to wash your hands, those are bacteria you want to remove. They're not necessarily bacteria you want to kill, and you don't necessarily want to kill everything on your hands all the time. So when you're considering what to stock in your home, you probably want to consider having a soap and not an antiseptic, which is also a reason to avoid that Triclosan from before. I know it keeps coming up. But if you are in a specific situation, healthcare situations, immunocompromised situations, then you might need to have an antiseptic or something that does at least inhibit bacterial growth instead of something that just washes off the ones that are coming along for a ride. But for your everyday use, soaps are almost definitely more than sufficient. All right, canning and botulism. So home canning can be a fantastic way to preserve food, but it can also be very dangerous if you're not careful, because Clostridium botulinum, we're back to Clostridia again, is a mostly obligate anaerobe, which means that it lives in low oxygen places. But when you can, a big part of what happens is you fill things up all the way, you make sure there's very little air. So a lot of these are vacuum sealed in such a way so the air is removed. That gives Clostridium a place to live. Then you need to sanitize the item in such a way so that if there is Clostridium in it, you kill it, except that Clostridium forms endospores. Endospores, if you'll recall, are these tiny little places where the bacteria can hibernate, and they have an extra coating around them. They are very, very secure. They're in fact so secure that boiling is insufficient to kill them. So if you take your cans and you vacuum seal them and then you just boil them, you might still have endospores in them, which means those Clostridia can go ahead, they can live in the food, and if they pop out, they might produce toxins that then survive. That's bad no matter what. However, what you really want to do, and as you can see in the second picture here, is pressure can. So that means you have a pressurized cooker. It brings the heat above boiling, and that can make it high enough to kill the endospores. So if you're going to can especially low acid foods, because they don't like acidity either, you definitely want to go ahead and have a pressure system for the sterilization process. All right. What do you think the best way is to clean your cutting board that you just used for raw chicken? I know it says how to clean raw chicken, but it's raw chicken like off a cutting board. You could wash it in very hot water, maybe even boiling water. You can pour boiling water on it, or maybe you could add some bleach to it. Well, boiling water definitely would help with the microbes, but it's not an immediate way to kill microbes, and typically you have to boil something for an extended period of time before it actually takes hold. So one of the problems with boiling is that if you only pour boiling water over something, it's not quite as useful. Bleach would definitely kill any of the microbes that are involved, and raw chicken especially is an issue because the Salmonella bacteria is a part of chicken flora and not our flora. So it's on or associated with almost all raw chicken products, but if it finds its way into our digestive system, it is automatically a pathogen. But of course, can you just dump bleach on things either? Well, you gotta be careful. Bleach is not an antiseptic, really. It does destroy human tissues. So if you're gonna use it on a cutting board, you gotta be careful about how much you're using, whether you're rinsing it off, making sure it's not getting on your future food, and of course, whether or not it's strong enough to destroy parts of that cutting board. Obviously, this doesn't have to be a one or the other type of choice. You can use a little bit of both. Definitely wash your cutting board well with soap, hot water, scrubbing as much as you can. And then if you're really concerned about it, or you're in a setting where it's important, like food delivery and things like that, you can use a dilute bleach solution to wipe something down and then make sure that it's cleaned off afterwards. So that can help things out. I know as a Girl Scout, I always learned the at least 3 bucket method of washing. So you had hot soapy water, you had cool rinse water, and then you had a 10% bleach solution. You dipped everything in just once, and then that way it was all set for the future. And my mess kits were almost always pretty clean. Freezing and thawing meat. This is something you're warned against a lot. You can freeze things and it will do a good job of keeping bacteria from growing on it. But you don't want to thaw out the meat and then refreeze it. How come? If the freezing is so good, then wouldn't it be good the second time? Well, so inherently, of course, the problem with freezing, it doesn't kill bacteria. It doesn't even really completely stop them from functioning. It just slows them down to the point where they're kind of hibernating. There are some bacteria that might die in the freezer, but most of them can survive fairly well. And they're not even really harmed by it. So the moment they come back up to even a semi-reasonable temperature, you know what they do? They immediately start dividing. They're still near food sources. They have plenty of moisture. They go ahead and they divide. So if you thaw the meat, you haven't stopped the bacteria at all. You just allow them to divide more. Then if you refreeze it, you're just freezing more bacteria. And of course, the dose of bacteria is important because it changes going forward. The next time through, if you unthaw it again, now you have more bacteria to begin with. They're going to double up again. You're going to have even more bacteria. So you can see why this is a problem. On the other hand, do always make sure you thoroughly cook all of your meats, because then you can kill all of your bacteria, because actual cooking heat, so beyond boiling, actual cooking heats that go well into the right ranges will often kill the bacteria pretty effectively. But also don't freeze and then rethaw if you can help it. Anthrax in the mail. So when I was in college, somebody sent some anthrax to a place in the mail. This is bad. Bacillus anthracis is another bacteria that has spores. I know, all these spore formers. There's so much trouble when you're dealing with control. They put them in envelopes and then people got sick from it, and this is an issue, and everyone went crazy. And people were like, well, what can we do to save ourselves and make sure this doesn't happen? We can irradiate the male. One of the things they were thinking was, well, we don't want to add too much radiation to the male, so what if we just put everything under a UV light? Would that work? Well, let's talk about radiation. So there's two kinds, ionizing and non-ionizing. Ionizing radiation might include something like gamma radiation, X-rays, things like that. Those go through materials and can actually change the atomic structures of some things, which means that, first of all, they are able to get through stuff, and they're able to destroy most bacteria, spores, things like that. Although they still can't get through everything. If you have a huge stack of something, it might be an issue. And if, for example, you've got something like metal in the middle, or, like you got a CD for something in the mail, that might cause a problem. But it could work. UV, on the other hand, is non-ionizing radiation, as you know, because you stand outside and it does not destroy you unless you're out there for way too long, and then it can cause minor problems. But if you put a stack of mail under UV, it'll take care of the surface, but not much else. Endospores might not even be killed by it, but they're definitely going to be difficult to kill going through something like multiple layers of paper. So although UV light was a nice idea because it was a little less dangerous, it's also not quite as useful. Although even ionizing radiation wasn't a great idea. And in all honesty, your mail is probably mostly safe. This was a pretty one-off type of thing, and that's not likely to happen again. But it was certainly an interesting experience in microbial control when it was happening, and people were trying to figure out the best way to control microbes through the mail. And honestly, there's a lot of mail every day handled by a lot of people. It's a pretty tough thing to control. Speaking of tough things to control, I'll leave you with one more microbial control sort of conundrum that people have been working on for, let's say, thousands of years and are still to this day, which is how to preserve food. I mean, we like food, but for example, I live in upstate New York. We get fresh food for only a portion of the year, and I'd like it to last longer than that. So what are the things we do to preserve food? There's a bunch of different options. All of them have pros and cons. When you're thinking about preserving, those are the things you should think about. So I have an example of dehydrated zucchini here. Dehydrating works for some foods and not others. And it does last pretty well as long as it's stored well without water. But of course, if I put something that's dehydrated on my counter in upstate New York, a lot of times it just sucks up the water from the air around it. So it's not always useful. But if I store it well in bags and I keep it out of the way, it can work. And some things are very delicious dehydrated, but others maybe not as much. You can freeze dry, which is a little bit better. So these are freeze-dried strawberries. That takes a lot of technology to make happen. And again, it only works with some foods. And then to eat them most of the time, like some freeze-dried things taste pretty good on their own. Freeze-dried strawberries actually are one of them. But freeze-dried like meats and cheeses and other things, you often need to rehydrate to eat. You can just freeze. I freeze berries all the time. I have a whole bunch of them in my freezer right now from essentially June and July. But you have to have freezer space. You have to keep them at temperature. To really get them to work, you should flash freeze, which is to immediately go to a very cold temperature. And that can be very hard to do. Companies often do it. Us as regular people can't do it as easily. And then, of course, if at any point you lose the temperature, you lose all of the items that are frozen. Another possibility includes curing and salting. So both of those change the water activity or add things that microbes don't like. And so these things last. So as you probably know, cured type meats, salamis, pepperonis last almost forever, although not entirely. And it doesn't completely save the actual structure of the meat product. So even though it might be able to keep bacteria out, it's going to change the way the meat product works, and it still doesn't last forever. So food preservation is an area of microbial control that's important. It is an area that honestly probably will always have jobs if you're looking for one, and is something that we're still working on. So microbial control has a lot to think about and a lot of areas in which there's still things going on. So in any situation, think about how you would control microbes. What are the best forms of control for your particular areas? What are the best forms of control to have around you at any given time? And how many microbes do you need to control? Always think about that. How clean does your area actually need to be versus how much does it need to be just to keep people from getting sick? And that changes day to day, year to year, person to person, and location to location. So lots to think about controlling microbes. I hope you learned and thought about some great things during this one.